Students, today we are going to discuss about the demonstration of high performance liquid chromatography, which is also called as HPLC. So HPLC is basically also called as high pressure liquid chromatography. So why we are using high pressure here? Because in the normal column chromatography, when we are running the samples and we are going to separate the samples, it needs more time duration. But in case of HPLC, when we are applying the high pressure, automatically the sample will be getting separated within a short span of time. So this is the reason we are preferring HPLC over the conventional column chromatography. Now coming to the instrumentation, how this instrument is basically working. So we have the instrument with HPLC Waters, right? So Waters is the company we are having, and here basically when we are having this thing, basically we are having this mobile phase. This is a solvent reservoir. This is the solvent reservoir where we are having the mobile phase, and this mobile phase will be pumped here and it is coming to the pump. The pump is, we are having two types of pump, that is upper one and the lower one. Two pumps are there. So these two pumps are basically the reciprocating pumps used in HPLC. So when we have the pump, basically we are using up to 5000 psi pressure, and the flow rate can be go up to 1000, that is up to 100 microliter we can go the flow rate. But generally here what we are doing, the flow rate here normally we are using is 1 ml per minute. Up to 10 ml per minute we can go. But normally for ideally the pressure should be 1265 psi for any unknown sample, and 1 ml per minute is the flow rate for most of the samples. So that we have to set this thing with the help of this pump. And here we have this valves also. We have the valves and we have the plunger also. So this thing will be coming and going as such. And here when the pressure is applied here, what the pressure, this high pressure will try to, this is connected here. You can see here the valves are connected here and it is coming over here. And this is connected over to this area where we are injecting the sample. When we are injecting the sample, basically what we have to do is, we should remember that we have the very important thing. This is called Hamilton syringe. There is a syringe called Hamilton syringe and needle. Using this syringe, the capacity of the syringe is around 25 microliter. Normally 10 microliter is sufficient for qualitative analysis. So what we are doing, we are taking the qualitative analysis of the sample. Before going for checking the sample, what we are doing, we just make the sample by dissolving in a suitable solvent. Pre-treatment we have to do. After dissolving the sample in a suitable solvent, then we have to use the membrane filter. So this is the membrane filter we are basically using. Basically the membrane filter size is 0.22 micrometer. New pore membrane filter having the diameter of 0.22 micrometer. Using this membrane filter, we just try to use this sample dissolved in a solvent, and then with the help of the membrane filter, we try to filter it out. If any of the particles are not dissolved, they will be filtered out, and then that pure sample solution will be injected into the column, right? So here this is an injector loop area. So in the injector loop area, if you look at here, basically in the load position only we have to inject the sample. In the load position only we have to inject the sample, right? After injecting the sample, then what we have to do is, after loading the sample here, then after that we have to just rotate here, so that the automatically the sample whatever getting injected, it will be entering into the column here, right? So this is how we are basically using this thing. So sample injection is another important thing. So all these things, why I am telling all these things, because it is a systematic flow. When we talk about the HPLC instrumentation, basically all the things can be controlled with the software itself. But one important aspect here, we can adjust the detector wavelength also. So when the sample, when we are running the sample and when it is passed into the column, so here the detector also will be glowing the brown light here. Instead of this thing, it will be glowing in a proper manner. Now what will happen, this column is very, very important. If you look at this column, the column is having the size of 4.6 millimeter into 250 millimeter internal diameter, and inside the column, this is basically the stainless steel column. Sometimes we can use a peak column for HPLC also. Peak column refers to polyether ether ketone, and this particular column is coated with octadicyl silane, because the silica gel is basically polar, and inside this column, basically this is a C18 column, which is also called as octadicyl silane column, where the polar stationary phase is clumped with some methyl groups and it becomes the non-polar stationary phase as such. And the particle size of the stationary phase octadicyl silane present inside the column is a 5 micrometer thickness. and this column is a long narrow column, and this will be more sufficient for carrying out the HPLC normally, right? And this column, that same column only here we have attached here. From the column, if you look at that, it is going into the detector. From the column, whatever it is going here, it is going to the detector. And from the detector, again, whatever the sample is running into the detector, we can adjust the wavelength of the sample here. Whichever wavelength we want to use it, we can use it. Basically, the detector, what we are having is UV visible variable wavelength detector. When we have this variable wavelength detector, we can scan the range from 200 to 800 nanometer. We can go up to 1000 nanometer also. So whatever the sample nature is there, accordingly we are using this UV visible detector. But there are certain cases where we can use the fixed wavelength detector, and advanced instruments, they will be having the photodiode array detector. Sometimes they have some other detectors like evaporative light scattering detector. So different detectors are used in HPLC, but we have this variable wavelength UV visible detector. So once we have this wavelength detector, when we adjust the wavelength here, automatically the sample will be detected with the help of the detector. And one more is also coming here. So this is basically the waste reservoir, where the rest of the solvents, whichever is coming out from the column, they will be coming out from the detector, and that will be passed into this waste solvent reservoir. So from the HPLC demonstration point of view, we should know that we have the solvent reservoir, which is attached with the pump. And the pump is basically having the reciprocating pump. We have the two pumps here, right, for better efficiency. From the pump, it is connected with this injector loop. The injector loop is basically made up of two things. One is load and inject position. Whenever we are loading the sample, we have to keep in the loading position. After that, that time when we are injecting the sample, then after that what will happen? When you rotate this in the clockwise direction, automatically the sample will run into the column. So column is the heart of the instrument. And the first part of the column acts as the pre-column, or simply we can call it as the guard column, which acts as a pre-filter, and the rest of the column acts as the original analytical column, which is used for separation. So the first part of the column is responsible for removing the unwanted particles absorbed onto the column, right? And after that we will be having the detector. Finally, the detector is very, very important, and the detector, UV visible detector, is used for identifying the sample, right? So this is all about the basic fundamentals of HPLC. As far as instrument is concerned, everything we can control with the help of the instrument itself here. If you look at here in the instrument, you can able to see here, I can show here there is a software we are using in the instrument. The name of the software is called Empower Pro software. So this Empower Pro software, we are having different columns here. One is run sample, browse project, configure system, process data, review data, and print data. Basically, whenever you want to run the sample for injection, we have to first click run samples. Then after running the samples, if you want to go for browse the project, then after that we go for review data. Only these three things we are using as such. In the browse project itself, we can process everything, and then finally we can review the data as such, right? So run samples, then we can go for browse the project, where we can go for process, review, then integrate, and then finally we go for preview publisher, where we can take the printout of the sample, right? And here, if you look at here in the file portion, if you look at here in the file, new sample method we can set, right? And then we can go for new method set, right? New sample set method template is also available. Whichever method if you want to set, accordingly you can set it. And inject also we can do in an automatic manner. Prepare the system for injection and make injection will be there. So this is all here. And sample name, what name we have to give, we can do it here. And then vial, what is the content here, that we have to only how many vials we are using, that also we can give here. Injection volume, generally we keep it for 10 microliter. And run time we can go for 10 minutes or 15 minutes. Maximum 15 to 20 minutes we can give for most of the samples, right? So this is for injecting the sample, and this is for that is loading the sample into the column, right? So this is how we are keeping. And here, if you look at here, we can have this thing. The detector is also, lamp is also in green, and detector also we can use what wavelength we are using, that also we can change it here. If you want to use 422 nanometer or some other wavelength, we can use it over here. And here the pump is having two things, that is flow A and C, right? So the pressure you can see 1435. High limit will be 4000 psi pressure. So depending upon the need, we can use the adjust the flow rate as well as the pressure in the system. So this is all about the various aspects in HPLC aspects. So this is very, very important. And remember that we can make the samples here. For example, the very important thing is that we can have the samples here. So any sample, whether it is paracetamol or aspirin, you choose different solvents. And after choosing different solvents, you just try to dissolve properly in solvent system and see which is having the best solubility. Only those sample dissolved in the solvent which is having high solubility, that only you have to take. And after taking the solvent, you just go for immediately try to go for solvent degassing or membrane filtration. We are using the membrane filter 0.22 micrometer membrane filter. Using that we can remove the undissolved particles, then we can perform the chromatography as. Remember that whatever solvents we are using, all the solvents whatever we are using here, they are all high quality HPLC grade solvents or spectroscopic grade solvents, right? So these solvents are having 99.9% purity, and these solvents before the solvents also contain some impurities which may be dissolved gases. You have to remove them just by going for ultrasonication or by going for membrane filtration or by passing through the vacuum, and then these solvents only will be kept over here and that will be attached to the pump here. Right? Similar case when the sample is also dissolved in solvent, the sample dissolved in solvent also has to be properly removed so that they should not clog the column. They should be removed with the undissolved particles. Then only you can go for the membrane filter and then you can pass the solution for injection. So I hope this is clear. Thank you very much, my dear students. Thank you very much.